overview of carbon cycling. Specifically, I'll be focusing on showing how different studies have evaluated the effects of plant biodiversity on ecosystem processes, and why trait-based approaches are important to predict carbon cycling. And at the end, and also we'll touch on how we're going to incorporate some new of these trait-based approaches to the activities that we're going to do in the course, and why we need to understand these new methods that we're going to use. So first, I just want to describe briefly, let's look at the carbon cycle. So we define the carbon cycle as, well, carbon interacts naturally between the ocean and the biosphere and the atmosphere. And throughout history, carbon has cycled naturally between these three environments and has remained relatively in balance. And here you can see that there are many different greenhouse gases, such as carbon dioxide, methane, and other gases, that they mix easily in the atmosphere and form kind of like a blanket that traps some of the radiation coming from the sun. And this blanket is what we know, or what is known as the greenhouse effect. And this blanket basically keeps the planet much warmer and allows for life to exist. And for more than 1,000 years, this natural carbon cycling has been in balance and keeping the concentration of CO2 in the atmosphere relatively constant. But after the Industrial Revolution, or with the beginning with the Industrial Revolution, humans have begun to rely more on fossil fuels, as you know, and that has released higher amounts of carbon to the atmosphere. So that has created an imbalance. As you can see here, this imbalance is basically shown by the red arrows. And this use of fossil fuels together with deforestation has offset the natural balance of carbon dioxide. Here we can see on the right part of the screen that over the past 150 years, the concentration of carbon dioxide has increased from 270 parts per million to 400 parts per million. And the higher the concentration it gets, the higher the greenhouse effect becomes. And as a result, we have the planet, we have a warming planet, as you can see here, also indicated by the red shadow. So when we look at the time series of the concentration of CO2 in the atmosphere, and this is data over time from the observatory in Hawaii. And you can see here that carbon dioxide has increased over time. And there are two different lines. So the black line represents basically the average increase over time. And in the red, you can see different ups and downs that is basically representing the seasonality of the planet between the southern and northern atmosphere. So there has been a lot of concern about what are the consequences of these increases of carbon dioxide on the ecosystems. And here, this diagram's a little complex, but what I want to show you in general is this is how we describe the carbon cycling, the carbon cycle globally. So now we have different numbers, but what I want you to focus specifically is in the values that are within the brackets that are in red. And it doesn't matter necessarily the amounts, but what you can see is that now all these reds represent the different anthropogenic fluxes. And this is the increase of these anthropogenic fluxes relative to what the fluxes were before the Industrial Revolution. So you can see that we have different sources contributing to increases of carbon dioxide that are actually influencing the maintenance of this balance of carbon cycle. Okay, And Brian?
Yep, I'm here.
Oh, sorry. So we leave the questions at the end, or the students are allowed to ask questions throughout?
Sure, if they want to ask a question right now, that's totally fine.
Oh, okay. Okay, I just want to make that clear. Okay, so since, so as I was showing here before, since the anthropogenic impact is so higher in terms of how much carbon dioxide we are emitting, different to due to the different sources, fires, fossil fuels, land use change, but also, yeah, all these anthropogenic disturbances. So because of this, there has been a lot of interest in understanding what are these anthropogenic impacts in the different components of the carbon cycle, and specifically how all these interact with the different drivers of global change. So since the beginning of the ecosystem ecology, since ecosystem ecology became a science, there was this framework that was developed to have an ecosystem approach to understand how global change drivers impact not only global cycle, but also other important components such as biodiversity loss. And here we can see one of the earliest papers of ecosystem ecology, and you can see the effect of human enterprises in the different global change drivers and how these affect biodiversity loss. However, here biodiversity was only considered a variable response, and we were only interested in evaluating the anthropogenic disturbances on biodiversity loss. But it wasn't until the beginning of the biodiversity and ecosystem functioning research that we started to think about how changes in biodiversity also impact ecosystem processes. Here, for example, you can see global changes are impacting biodiversity, and biodiversity at the same time, due to changes in species traits, can have a direct effect on ecosystem processes, but also an indirect effect on the provision of ecosystem goods and services. But unfortunately, the way that changes in biodiversity have been evaluated to understand how carbon cycling responds across the atmosphere has been used plant functional types. So basically, the approach that has been used from Earth and climate scientists to understand how vegetation is changing across the globe relative to different climatic gradients and how those changes impact carbon cycling. They look at changes in vegetation cover mostly, based in classifications using biomes, different biomes. So you can see here, for example, they use how vegetation cover was in the past, and they try to project these changes into the future based on how vegetation is affected by different climatic gradients. And as a result, we can have projections of how the vegetation will change over time. But you can see here that the confidence in these projections is very variable, with many regions showing high uncertainty in the models of how vegetation is going to change into the future. And the reason of this high uncertainty is because so climate scientists and earth system scientists are failing to incorporate different variation in vegetation. For example, these models lack of incorporating vegetation across heterogeneous landscapes. We know that vegetation types have variation that is horizontal, but there is also vertical variation that it's important to account for. So for example, this picture shows how the same species can have different variations in leaf size across the vertical gradient, as you can see here. And these different leaf size are going to affect the different rates of carbon capture in different individual plants. But this variation is rarely incorporated in models to predict how global carbon cycle is going to change into the future. So we use a trait-based approach. I think Brian mentioned in his class what are the importance of trait-based approaches, specifically to understand variations across the species. So here, I just want to recall the definition again. We use a trait-based approach to understand all the variation among plant communities and how this variation impacts rates of carbon capture. So for example, leaf size is a functional trait that is related to growth, and the leaf size is gonna be affecting rates of photosynthesis, for example. So to understand how all these different variation among plants affect ecosystem processes, we study how these variation in traits affect photosynthesis, which is the most fundamental ecosystem processes related to carbon capture. So photosynthesis by plants provide the carbon and energy that drive most biological processes and ecosystems. And I'm gonna show you how photosynthesis affect the carbon cycle within the plant. So if you can see here in this diagram, carbon enters the ecosystem as photosynthesis or gross primary productivity. And this photosynthesis, so carbon enters the plant via photosynthesis, and this photosynthesis goes, it's called gross primary productivity. And it goes to the different compartments in the plant, to the roots and to the soil. But there are other different carbon inputs within the plant that provide, that contribute to the carbon cycling. So for example, net primary productivity that accounts not only for the amount of carbon that is captured by the plant, but also the amount of carbon that is released through plant respiration. And we also need to account for the soil respiration or for the respiration of other organisms. So here, this diagram shows better how the different carbon inputs from the different plants and the different fluxes. So most carbon gain is captured by gross primary productivity of photosynthesis. As you can see here, all the carbon goes to the plant for plant growth, but it also goes to the roots. And once it enters the soil, there are different amounts of the carbon that is also released, via respiration by microorganisms, as you can see here on the right with the microbes. But there is also respiration by the different compartments of the plant. So not only the leaves, but also the stems and the roots. So when we take into account all the different fluxes that are coming in and out of the plant, then we can have an estimation of net ecosystem exchange. And you can see here that basically net ecosystem exchange is defined by gross primary productivity, or the amount of carbon captured by photosynthesis, minus respiration. And this respiration includes respiration by plants and respiration by animals, or respiration by microorganisms. And when we only consider the respiration by plants in these equations, then we call this process net primary productivity, or you can see here on the left. So these are one of the carbon pools and fluxes that we will be measuring in the course. And I just want to show you how these fluxes vary at individual level. And here you can see this is from one of the book chapters that you have in the folder for the lectures. This is basically what are the main factors governing variation in gross primary productivity at ecosystem level. And you can see here, but there are different controls that are split in long-term controls and short-term controls. And most of the state controls that are here on the left are related to environmental factors or abiotic conditions. And as I was mentioning before, most of these factors are actually used in Earth system models, and plant functional types are also used in Earth system models. But as you can see here, These different arrows represent the importance of each of these factors and how they affect the gross primary productivity. So basically, the large the arrow or the thickness of the arrow represent the strength of the effect and how this attribute is important in controlling rates of carbon capture. And you can see here that leaf area, nitrogen, and season length are very important for carbon capturing, but these are most of the attributes that are actually excluded from Earth system models, but that we are actually incorporating by using trait-based approaches. As you can see here, for example, leaf area is very important, and variation in leaf area is going to have an important effect on how much carbon is captured by the different plants, but also season length of variation, temporal variation is gonna have an important, is gonna be an important driver of how much carbon can be captured as well. And another important driver in how plant communities capture carbon, or basically in the rates of carbon capture among different communities, it can be explained by the slow and fast continuum, or the leaf economic spectrum. And you guys also saw a little bit of this from the previous lecture by Brian. You saw, for example, how the different leaf economic spectrum explain variation among plant communities and among these different traits, but how these traits impact the rates of carbon capture. So if you can see here on the left, we have plant communities dominated by slow growth or low tissue concentration. And when you have communities dominated by the slow growth rates, these communities are gonna be very good at storing carbon over time. For example, they will have low ecosystem production efficiency, or they will also accumulate litter over time and will have a very slow turnover rate. And as a result, they will be better at storing higher amounts of carbon in the long term. In contrast, if you see the fast growth communities, you can see that there is a high difference when you have plants that are that have rapid carbon and nitrogen turnover. So these are communities that are dominated by high turnover rates, high productivity, high rates of litter decomposition, and as a result, they will have a high rate of carbon lost. And understanding how these different communities impact the amounts of, or the rates of carbon capture is very important, because it's gonna affect also the different plant and atmosphere feedbacks. So for example, in terms of feedbacks, fast growth communities are gonna have a negative feedback in terms of the amount of carbon dioxide that they can store from the atmosphere. And in contrast, slow growth communities, because they have a slow rate of accumulation over time, they're gonna have a positive feedback on the atmosphere because they're gonna store higher amounts of carbon. Okay. So there have been two different mechanisms, or there have been different mechanisms hypothesized to explain how plant effects are going to impact ecosystem processes such as carbon cycling. And here, these are summarized. I'm sorry, there's a mistake here. Okay. So these different mechanisms are called the mass ratio hypothesis or the niche complementarity hypothesis. And I'm gonna explain how that works here. So if you have a species pool and you sample the different community, you can have different variation if you're in your community that is represented from trait variation of the most dominant species on the left part of the diagram towards higher trait diversity that it tends to be correlated with higher species richness. So these different mechanisms of how plants will affect ecosystem processes are going to change depending on whether, for example, carbon capture is dominated or is mostly explained by the dominance of a species with particular traits, or whether rates of ecosystem processes such as carbon cycling is actually explained by the higher trait diversity among the species, or a mechanism that we call niche complementarity. And these two mechanisms are not completely excludent. You can have both in a similar, in the same community. And as you can see here in the diagram, it's kind of like a continuous variation among communities. And this diagram actually highlights the importance to not only measure species abundance, but also the intraspecific and also the interspecific variability among traits, to be able to disentangle the importance of whether the mass ratio hypothesis that is related with the dominance of a species with particular traits is probably the mechanism that is explaining more changes in one ecosystem processes, or whether these processes are strongly explained by niche complementarity. So I just put this diagram in terms and try to explain what are we measuring, or how can we link the intraspecific variation in the other groups and link these to the different carbon fluxes that we're gonna measure in our group. Okay, so now I'm just gonna show you different studies where we have evidence of different diversity indices on different carbon pools and fluxes. So this study shows, this was one of the most famous experiments and one of the first studies that show that primary productivity in grasslands actually increase with the number of species. And this is one of the experiments in Cedar Creek. You might know it for all the papers by David Tillman. I just decided to show this because here we have different components of biodiversity. As you can see, this study shows that the average productivity in grasslands not only increased with the number of species richness, but it also increased with functional groups and with different metrics of functional diversity. And these functional diversity metrics can be estimated from the different traits within the community. So this is important not only to highlight that it's not only the species richness that it's important, but there are also other components of biodiversity that are able to explain different rates in carbon gain. And here I want to mention different studies. I think one of the first studies also within the literature that was recommended. These are one of the classic studies that have shown how rates on different pools of carbon. So the first one shows increases in the community weighted mean with primary productivity. And this one is a synthesis that shows that not only we have increases in productivity thanks to all the experiments that have been conducted, but this is also a result that has been supported by many different field studies. And these two are other specific studies that show contributions of traits and how the traits impact different ecosystem processes, not only above ground, but also below ground. And I'm gonna put the references of these when I upload the lecture. But I didn't wanna spend too much time showing all this evidence that, because you have a chapter where you can read the synthesis. But what I wanna emphasize mostly is why we need to focus on the traits and how can we learn from the different traits in terms of predicting rates of carbon cycling. So this diagram is, if you can see here, this comes from the book chapter from Garnier. You have it in your folder. But you can see here, again, different change, how different traits at different scale impacts the rates of carbon gain. So on this first diagram on the left, you have the net photosynthetic rate and how that changes with a specific leaf area. And you can see here that there is a strong positive relationship. And we can have more data because we are working at a smaller scale. But you can see that this positive relationship in the amount of photosynthetic rate or the amount of carbon rate that is captured by the plant is still the same, even if you measure the relative growth rate at the level of the whole plant, as you can see here on the bottom. Or if you look on the right panel, you can find the same positive relationship when we measure the amount of carbon gain or net primary productivity at the whole community level. And I think this diagram is very important because what we're gonna do in the course is that we're gonna measure trace at leaf level, and we're gonna actually measure trace at whole plant level, specifically in our group, but I'll talk about that in a second. And this community level on the right basically will represent the community weighted mean that we can calculate from using all these traits per plot. So you can see here that these relationships are not only the same across different levels, but also that by incorporating the leaf scale, we can actually have more data and also capture more of the variation across the different species. So I wanted to show this diagram that is one of the most recent studies of how tree species richness affects carbon fluxes and pools, just to try to show you how different compartments affect carbon fluxes. So I know this diagram is a little complex, but what I want you to focus on is that basically each of these boxes represent different pools and fluxes in an individual plant or in an ecosystem, where you have AGC or all the different Cs represent either carbon pools or fluxes. And of course, the ones that are above the dash line represent above ground pools and fluxes. I'm sorry. And the ones that are below the dash line represent below ground fluxes. And I wanted to show you this because this is something that also has been neglected from the literature, that trait-based ecology tend to focus on understanding more about ground fluxes than below ground fluxes. But there are few studies that have attempted to disentangle the importance, in this case, of tree species richness and how that affects the different carbon pools and fluxes. So basically, each box represents a carbon pool, and the red arrow represents whether there is a positive effect of a tree species richness in the carbon pool, and the blue represents whether there is a positive effect in stand age. And this is successional age, or basically is age since last disturbance. This is from a chronosequence study. So you can see that pretty much in every carbon pools and fluxes, you find not only an effect of the different, of the species richness. So species richness was positive, had positive effects not only in above ground carbon pools, but also in below ground pools and fluxes. But we also have an effect of an stand age. And this is something that we will try kind of measure similar stuff with the different fluxes that we're gonna do in the different Born plots, because the Born plots, as far as I understand, they are in a chronosequence. So we will be able to disentangle the effect of different traits, in our case, and the different age or different chronosequence treatments on the different fluxes. Okay, so coming back to the importance of evaluating the scale, as I showed you before, we found positive effects of some leaf traits, specifically a specific leaf area, on different rates of carbon gain at leaf level, at the individual level, at the whole plant level, and also the community level. So the importance of measuring these relationships at different levels is in order to develop methods that allow us to scale up from individuals to communities and ecosystems and improve predictions of carbon cycling. Most of the models that are currently developed by Earth system modelers or climate scientists are actually attempting to develop, basically to use ecological scaling rules to parameterize models. There are different ways that we can develop these ecological scaling rules. And I think Brian mentioned one of these, which is the trade driver theory. Also, Sean mentioned something related to metabolic scaling theory. So one of the things we wanted to do in this group 4, in order to try to develop scaling rules, is that we're gonna measure the fluxes at community level in each plot, but also at the whole plant level. And this is one of the projects we're gonna conduct in the course. So here I'm just showing you pictures of the similar measurements that we're gonna take in Peru, but this is from Ramble in Colorado. If you're not familiar with the method or haven't checked the papers on the method that we use, this is the picture representing the flux stem method. Basically, we will use a Lycor 7500 that is kind of like a tiny flux tower. And we build this stem around the sensor head to measure photosynthesis. And we will measure respiration using a dark tarp, as you can see on the right. So we will measure photosynthesis and respiration at ecosystem level. And by doing so, we will be able to calculate net ecosystem exchange, net primary productivity, gross primary productivity, and ecosystem respiration. And we are also going to measure soil fluxes, sorry, not soil organic carbon, but soil fluxes using a different device, not the one that is here, but we will measure soil respiration to be able to calculate the net ecosystem exchange. And what is new in terms of the project for this group is that we are very interested also in measuring respiration at the individual level. And as I mentioned, we wanna measure things at individual, but also at the leaf level to be able to scale up from individuals to communities and ecosystems. And specifically, if you remember from this diagram, we have different compartments contributing to different carbon budgets. So we will be measuring photosynthesis at the plot level, and we will be measuring traits related with photosynthesis at the leaf level. But we also wanted to measure leaf respiration or plant respiration. We want to estimate plant respiration for the most dominant species within the plot. And for this, what we're gonna do is that we're gonna collect some individuals and we're gonna measure the leaf respiration. And the way we're gonna do this is, well, first, we wanted to address a particular question that is whether mitochondrial activity really drives ecosystem respiration. So basically, we want to measure respiration at the leaf level and attempt to understand whether rates of respiration at the leaf level correspond to the different rates of ecosystem respiration or not. Because by understanding how this process changes across the scales, we will be able to understand whether we can predict these different ecosystem processes using leaf level measurements, and whether these relationships are also the same at individual level and an ecosystem level. So basically what we wanted to do by measuring leaf respiration is kind of building these different plots, but instead of measured net photosynthetic rate as we measure here, which is basically the amount of carbon gain, we will be building these similar plots but measuring respiration. So we can actually have an idea of the total net balance at the different three scales. Okay. So for the next assignment, I'm gonna give you more literature about this so you can have a better understanding on how this works. But basically the way we measure leaf respiration is, well, remember that plant respiration happens essentially within the mitochondria here on the left. And the mitochondria uses oxygen to activate the electron transport change between the carbon dioxide and the oxygen, and with the internal membrane of the mitochondria in order to produce ATP. So the electrons that are used for ATP production ultimately come from high energetic carbon compounds called photosynthetes. And these photosynthetes are being produced by assimilating atmospheric CO2. And when photosynthase oxidate in respiration, carbon dioxide is produced and ultimately goes to the atmosphere. So we are gonna use a technique where we actually color the different leaves, and we'll show you how this works in the field. I don't wanna go into the technical details right now, but basically what we're gonna do is measuring this leaf carbon balance in the most dominant species. And we will attempt to determine whether these variations at leaf level can correspond to variations of ecosystem level. And if so, if we can have this correspondence, then we will be able to have a better models to predict not only the amount of carbon gain, but also the amount of carbon that is lost by the different communities. Okay, so in summary, I show you how we can study differences in plant composition and diversity to understand processes in carbon cycling. I also show you how trait-based approaches provide a mechanistic link to plant attributes, not only to understand carbon capture and gain, but also to understand carbon losses via respiration. And I'll show you how we can, we need to use trait-based approaches to develop scale-up methods that allow us to predict how carbon cycling is gonna change in terrestrial ecosystems. So with that, I'll be happy to take any questions.
Thank you so much, Sandra.
Thank you, Brian. I think it's only you and me.